Hello, everyone, and welcome to today's webinar, Expert Insights, the growing role of genetic analysis tools for cell therapy development. I'm Christina Bowens, Global Market Development Manager for Digital PCR at Thermo Fisher Scientific, and I'll be your moderator for today's event. Today's educational web seminar is presented by Labroots and brought to you by Thermo Fisher Scientific. To learn more, visit www.thermofisher.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. Your questions will be addressed by the speakers via the contact information you provided at the time of registration. You may also submit any technical issues here as well, or if you're having trouble hearing or seeing the presentation. With that, I'd like to introduce our speakers. First, we have Dr. Haixia Wang. Dr. Wang is a director at Arsenal Bio as the head of analytical development. She is leading a team and has established multiple analytical methods for Arsenal Bio CAR-T product release and product characterization. Before she joined Arsenal Bio, she worked on allogenic CAR-T product development at Pfizer and Allogene Therapeutics. And next we have Dr. Wendy Nee. Dr. Nee holds a Ph.D. in biochemistry and has decades of experience in the fields of pathogen detection, cancer research, and cell therapy. Currently, Dr. Nee is the principal scientist of analytical development at Arsenal Biosciences, where she leads the molecular biology group developing assays using real-time and digital PCR technologies. These assays are being used to characterize CAR-T products that target solid tumors. Please see the presenter tab for complete biographies from our speakers. And with that, we'll begin the event. Hello and welcome to today's event. I'm honored to be your moderator for this discussion and the role of genetic analysis tools in cell and gene therapy development. We have a distinguished panel of experts from Arsenal Biosciences analytical development team who will be sharing their insights and expertise with us today. Thank you both for being here. So to begin our conversation, let's start with an overview of your typical workflow where genetic analysis tools like digital PCR and qPCR fit into this process. Dr. Wang, could you provide us with some insights?
Sure. Yeah. First, thank you for inviting us to this event. I'm Haixia Wang. I'm the director of analytical development team in Arsenal Bio. Right now, Arsenal Bio is a clinical stage cell therapy company. We're working on very advanced CAR-T product development targeting solid tumor. Our first clinical program, AB1015, is autologous CAR-T product for treatment of ovarian cancer. So our cell products are generated using non-virus system, CRISPR-mediated single DNA cut site integrated in site-specific site in the genome. So in order to test our product, we develop a bunch of analytical assays. Digital PCR is our main tool to characterize our cell product for reported integration efficacy, the transgene integration efficacy to the target specific genome site.
Great, thank you so much, Dr. Wang, for that overview. And so to dive a little bit deeper, can we explore the advantages that these tools offer? I wonder, Dr. Nee, could you describe where you find the main advantages of using tools like digital PCR within these workflows are and how they compare to other quantitation methods?
Oh, yeah, sure. Hi, I'm Wendy Nee. I'm the principal scientist of analytical development at Arsenal Bio. Leading the analytical molecular biology assay development using mainly the real-time PCR and digital PCR technologies. Yeah, that's a good question. So digital PCR has become very popular in recent years, especially in the cell gene therapy field. It offers several key advantages compared to other quantitation methods. We can list a few of them. For example, dPCR can do absolute quantification of target molecules. It does not need any standard curves or reference samples. Digital PCR also has much higher sensitivity compared to other quantitation methods. It can detect and quantify targets at a very low levels, can down to several digits. Other advantages like increased precision. because in digital PCR, each sample is divided into thousands of individual reactions, which provides a much higher level of statistical confidence. And of course, the digital PCR offered enhanced accuracy and robustness compared to like qPCR. So the digital PCR results are less affected by the PCR amplification efficiencies, inhibition, or sample impurities. So the reason behind is that the software can only need a yes or no answer. The fluorescent intensity does not matter that much. Yeah, here are just a view. So yeah, dPCR has many advantages over the other quantitation methods.
All right, thank you so much. So now to talk a little bit more about both qPCR and digital PCR technologies, since you mentioned you leverage both real-time and digital PCR for your workflows. Can you talk a little bit to how these complement each other and what factors might contribute to your selection of each technology? I wonder if Dr. Wang, you might take this one.
Sure, yeah, I can answer this one. So as I mentioned that in our product analytical platform, we use multiple technologies. We use both qPCR and digital PCR. It totally depends on the specific needs for our application. How to choose that depends on several factors, right? For example, qPCR is often used to report relatively quantitation, like target gene expression measured relative to the reference gene or control sample. In contrast, digital PCR enables the absolute quantification by directly counting the copy numbers, in the sample. So you don't need to run standard curve, for example, right? A well-established digital PCR-based method could be much more sensitive and precise compared to qPCR. It's particularly useful for detecting very low abundant targets, rare genetic variants, for example, or small fold change in gene expression. But currently, the cost of digital PCR is still relatively higher than qPCR, which is more cost effective. So depending on the different specific needs, you can consider those factors for choosing either qPCR or digital PCR. Yeah, that's our thoughts.
Yeah, thank you very much for that explanation. So as we explore the impact of digital PCR on cell therapy research and development, can you discuss the implementation of these workflows and the impact that they have on efficiency and reliability? I wonder, Dr. Ni, would you like to share your insights?
Yeah, I can answer this question. As we mentioned earlier, there are many benefits the digital PCR technology can offer. Overall, I would say the digital PCR has been a great addition to our molecular biology toolbox for both the research and the development processes. It allows us to do precise measurement of gene expression, quantification of the gene editing efficiency, and detection of very rare genetic variants. All this provides a more comprehensive view for the underlying biology or mechanisms. Regarding the automation, yeah, definitely this would be a huge benefit for high throughput applications. We all know that the automation reduces the hands-on time, so researchers can focus on other critical tasks. And it also minimizes the risk of human errors and operator-dependent variations. All these are so important for data reliability of the research and development processes. Yeah, that's it.
That's great. Yeah, thank you so much. And actually to continue on that line a little bit, I was wondering if we could discuss your thoughts on best practices around optimizing sample processing so that you ensure that you get accurate and reproducible results, from day to day or from run to run and experiment to experiment. Dr. Wang, could you provide us with some of your insights as to what best practices we might want to consider?
Yeah, that's a good question. Maybe let Dr. Nier to answer first.
Oh yeah, so the best practices in digital PCR, right? Yeah, there are quite a few of them. So maybe Haixia and I we can help this question. I can start first. Number one is that regarding the sample preparation, it's very important to establish and follow SOPs to minimize the variability, and also, if possible, implement appropriate controls to monitor the sample quality and integrity throughout the workflow. Number two is regarding the sample quantity and concentration. That's another very critical factors to be optimized. The amount and the concentration of the DNA and the RNA used in your digital PCR. So basically, too much or too little input can affect the sensitivity, accuracy, and precision of the digital PCR results. Maybe Haixia, anything else you would like to add to this question?
Sure. Yeah, this is a good question. In order to have a very accurate and consistent results in our group, we always remind our team member to have a good practice in documentation. For example, the lab records must be maintained in a very comprehensive documentation, including all the sampling, processing steps, and the data analysis workflow. So to get very consistent results, and that could facilitate if they have some trouble or some failed results, right? It also can support the future regulatory compliance. So based on the results during assay development or the feedback from QC, we always transfer the assay to QC to support the GMP testing, right? So we need to continuously improve the method by identifying some areas for improvement and then implement some corrective actions if there is a need. Of course, all of those good practice is applicable to all the analytical assays. So by following those good practice, you can optimize the not only sample processing in digital PCR, right, in all the assays even, and the leading to have a more accurate and reliable results that are very critical for cell product development.
Yeah, that's great. Thank you both for highlighting all of those best practices. I think they'll be really valuable. And so thinking a little bit about one line that you mentioned, can you discuss any considerations that one might take looking at regulatory guidelines related to the use of newer technologies like digital PCR when you're in the process of doing cell therapy research and development?
Yeah, sure. I think I can share some comments for this. So using digital PCR technology in QC tests, we definitely need to pay extra attention to regulatory considerations and guidelines in order to ensure the accuracy and the consistency of the results. Yeah, that's very critical for testing or characterize our product, like the safety, efficacy, and quality, right? This topic could be pretty long, but we can check the agency's guidelines as a reference. But keep in mind, the specific regulations may vary by countries or regions or by time. So generally speaking, develop or optimize digital PCR assays. We should, follow the agency's guideline, best practice, right? We need to have the documentation or standardize the data analysis workflow to make sure everything just, have the compliance. Another part like documentation, data integrity, how to, whenever we generate some data from digital PCR, we have to maintain, the full site or documentation that could be used for assay development or, optimization, assay improvement, or the future assay validation for later phase. For any PCR related assay development, reference standards, controls are really, really critical. So we will highly suggest to think about it, what kind of material could be used as a standard or as a material, as a controls, and negative controls, positive controls. Build after those controls early, that will be definitely beneficial for assay development. In our analytical development group, our main goal is developing the assay, and it will transfer to QC to support the GMP product manufacturing, right? So at the early phase, we need to at least qualify the release assays, product release assay. At the late phase, we need to get the assay validated. So for assay validation qualification, we should check the agency's guideline, and then to go through, which parameter we need to carefully check. It depends on different assays, case by case, to design the protocol for qualification or validation. Yeah. Whenever you establish some assays like digital PCR assays, we have to evaluate, the protocol, and then gather all the data and run some deep analysis to see is there some risk or what's the mitigation plan, especially if you want to apply this digital PCR-based method for late phase study. When I say late phase, that means, your product may be ready for BLA or commercialization. So that's really, really important, to think about it, what's the regulatory requirement, right? It could be, evolve over time, and then some specific guidelines maybe vary, depends on the stage of your product development. Yes, for example, preclinical or phase 1, phase 2, or commercialization, right? So it's very essential to engage with some regulatory experts and consult some relevant guidelines, and then communicate with the agency to make sure, all the assays meet compliance, and then keep updated, make sure all the requirements from the agency. Yeah, that's pretty much from me.
Yeah, great. Thank you so much for emphasizing the importance of really adhering to these high standards for analytical evaluation and to transfer into regulatory compliance. So we're kind of nearing the end of the discussion. So I wanted to get Dr. Ni, your insights into, some of the limitations or potential pitfalls of using digital PCR in cell therapy development, and how you might mitigate some of those challenges. Really to see, how do you stay updated with the latest advancements or best practices for these types of applications?
Yeah, that's a good question. So while the digital PCR offers many advantages for the gene therapy development, there are for sure some limitations and potential pitfalls that need to be considered. For example, the cost. So digital PCR can be more expensive than other PCR methods, such as the qPCR. So it's recommended to consider like cost-benefit analysis to determine if the increased precision and the sensitivity and accuracy from dPCR outweigh the additional expenses. Another challenge is that digital PCR requires specialized technical expertise in the assay design, optimization, and data analysis. Developing the robust and specific assays can be very challenging, especially considering the need for multiplexing and the presence of complex genetic modifications, for example. To these limitations, here are some suggestions or ideas. Collaborate with experts in the dPCR and or molecular biology community, and attend some workshops, training programs, or conferences focusing on the digital PCR or the cell gene therapy topics to enhance the technical expertise. Has there any other limitations you can add on to this question?
I think similar to other assays, right? If some assay we develop, we are going to use it in the QC lab, we always ask how to standardize the procedure, how to standardize the data analysis and the data, the final results report, how to report, right? So if for the whole process, if we could, come up with a more standardized protocol and then put a very clear guidelines for QC, so that will definitely beneficial for IC establishment, yeah. And then another thing, like as I mentioned, the data analysis for digital PCR data analysis definitely rely on the software, right? It could be a little bit complex. So proper data analysis techniques or a good algorithm that could be very essential. If we, like Thermo Fisher, if you can provide validated software tools, that could be very beneficial for us to implement those assay for QC and GMP lab use. Yeah. Anything else? I think that's much from me. That's pretty much from me. Wendy, do you have some add-on?
How do we stay updated with the latest advancements? Is that another question?
I think you already answered that one as part of your response, so that's perfect.
Good.
Yeah, so to wrap and close, I wanted to ask our final question here. So I'd love to hear your thoughts on the future implications of genetic analysis tools for analytical evaluation in your space. So I'll open this up to you both for some kind of closing thoughts on the future outlook.
Yeah, sure. Yeah, I think overall digital PCR is very useful for us to establish some assays where it can report very precise, very consistent results. And then it's also a QC-friendly method we would like to use. So for future, let's say, if we can further enhance the IC sensitivity precision, that could, make this tool more ideally for like identifying or quantifying some rare cell populations or rare gene events. That could be provided, more valuable insights for future use. Digital PCR definitely could be used for characterizing some genetic modification. So many cell products, they are using actually gene editing technologies, right? For example, CRISPR, right? So digital PCR can be used to accurately measure the frequency of gene-specific modification or, genome modification or gene expression, yeah. So talking about, improve the cell product efficacy or persistence, right? So if the digital PCR could give us some single cell resolution for cell product characterization, they're definitely will very beneficial for cell product development. Yeah, not only cell products, gene therapy is also very useful. So any other thoughts? I think using digital PCR for product quality control or to ensure batch consistency, this tool could be a very useful tool. We can think about what kind of assays or molecular biology-based assay, but this could use a digital PCR to report some valuable attributes. Yeah, that would be beneficial. For gene therapy product or cell therapy products, we always care about some impurity testing because we use multiple kinds of materials, raw materials or starting materials for gene product or cell product manufacturing, right? So we need to double check whether there are some leftover materials related to impurity or whether there are some contamination or other things, right? So I believe a digital PCR could be very beneficial to answer those questions as, because digital PCR it looks, much more sensitive and accurate. Yeah. So I think that's pretty much I want to share. Wendy, do you want to add some?
Maybe I can add a little bit regarding the potential innovations in the digital PCR, field. Yeah, it's a little bit hard question, but it's always my favorite to think about the future innovations. That may further shape the field of the gene cell therapy. Here are a couple I can think about. It would be ideal to have a higher capacities for multiplexing. I know currently it's up to 5, maybe even more for the multiplexing purposes. Another thing is that it would be great to integrate with next generation sequencing or next single cell analysis. That would be great. Maybe real-time monitoring. Let's see what's the future digital PCR will be.
Absolutely. Thank you so much for sharing your vision of future implications in this space. So again, I wanted to thank everyone for joining us in this really insightful discussion on the role of genetic analysis tools in cell therapy research. We really gained some valuable insights from our panelists here at Arsenal Biosciences. And before we conclude, I'd like to extend a special thank you to both Dr. Wang and Dr. Ni for sharing your expertise and perspectives with us, as well as your time. And thank you to our audience for joining.
Yeah, thank you for inviting us. Yeah.
Yeah, awesome. Thank you for having us. Yeah, thanks.
Thank you again, Dr. Wang and Dr. Ni, for your time today and your important research. We'd also like to thank Labroots and Thermo Fisher Scientific for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today. Your questions will be addressed by the speaker via the contact information you provided at the time of registration. Those questions submitted during the on-demand period will also be addressed by the speakers via the contact information you provided. This webcast can be viewed on demand, and Labroots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Goodbye.